Growth cycle or reproduction in bacteria occurs by a process known as binary fission. This is a process whereby a one bacteria cell is converted into two bacteria or two daughter cells. So there are various steps of this binary fission. You can see here is the cell having cell wall, cell membrane, and this is the elongated nucleoid. In the next step, this elongated nucleoid will divide and the transverse septum is formed from the cell membrane and cell wall in vagination. In the next step, the transverse septum completely divide the cell and then you have the two daughter cells from the one cell. This is the binary fission process whereby a one bacteria has given rise to two daughter cells. So then there is a concept of exponential growth whereby, as we discussed, one bacteria give rise to two bacteria cells, so 2 will give rise to 4 and 4 will give rise to 8. So that's the process whereby the basic formula is 2 to power n. So after first generation, you can see in the exponential growth pattern, you will have 2 bacteria after the first generation. After the second, you will have 4. After 3, you will have 8. And after 4 generation, you will have 16 bacteria. So this is the exponential growth pattern of bacterial division. And finally, we can see the division time of the bacteria. This varies. For example, E. coli can divide in about 30 minutes. And then there are some bacteria, for example, Mycobacterium tuberculosis, which can take about 24 hours to divide. This graph presents the growth cycle curve of bacteria, which shows that the bacterial growth cycle or life cycle has 4 major phases. In the first phase, we have the lag phase. This A presents the lag phase, where the bacteria simply prepare for the division. They do not divide actually in this phase. In the next phase, we have the log phase. In this phase, there is exponential growth. In this means that it's the log phase where the division of bacterial cells occur. In the next step, we have the stationary phase, which is presented by the C here. And this stationary phase, what happens that the nutrients start to deplete and or the toxic products accumulate and the number of bacteria some of them start to die, so that the number of bacteria which are reproduced or dividing are equal to the number of bacteria which are dying. This is the stationary phase. And then we have the death phase, where the number of dying bacteria becomes greater than the number of bacteria which are being produced. So the bacterial growth curve start to go toward the decline phase, where number of dying bacteria becomes greater than the dividing bacteria. So finally we can summarize the growth cycle curve, which means that it comprises of lag phase, which is the preparatory phase. As we said, no division of cell occurs. Then we have the log phase, where you have the division phase of the bacteria, and this is the exponential growth phase. And then we have the stationary phase, where the number of bacteria that are dividing becomes equal to the number of bacteria that are dying. And finally we have the death phase of bacteria, where the bacterial population start to decrease. That is, the number of bacteria which are dying become greater than the number of bacteria which are produced, and the graph goes toward the decline phase as shown by the D. And so this is the basic 4 steps of the growth cycle curve.